In this animated tutorial, we will be covering the basic elements of the Renin-Angiotensin-Aldosterone system, otherwise known as RAS. RAS is a hormonal cascade signaling pathway responsible for homeostatic control of arterial blood pressure, tissue perfusion, and extracellular volume. In order to obtain a better understanding of the pathway, we will first cover the structure and function of key elements involved in the system, including the kidney, nephrons, and glomerulus. The kidney has a functional role in the regulation of blood volume and blood pressure by altering the loss of water volume in urine, releasing renin, and releasing erythropoietin. Other functions of the kidney include regulation of plasma ion levels, such as sodium, potassium, and chloride, by adjusting the amount of such ions lost in urine, control of calcium levels due to calcitrol production, and stabilization of blood pH by controlling loss of hydrogen and bicarbonate ions in the urine. The kidney also plays a role in nutrient conservation by the elimination of waste products such as urea and uric acid and excretion prevention of required nutrients. Finally, the kidney assists in detoxification of poisons and deamination of amino acids, which allows them to be broken down by other tissues in states of starvation. These functions hold a high metabolic demand, and so the kidneys receive approximately 25% of cardiac output. The kidney has three major structural divisions within a protective fibrous renal capsule. The outer layer, shown here, is the renal cortex. The middle layer, shown here, is the renal medulla. The highlighted central zone is an inner pelvic region. Within the medulla, there are 6 to 18 renal pyramids, separated by renal columns. The renal columns provide blood flow to the renal cortex. Running through the renal pyramids, then through the renal papilla, small tubules are present which carry urine. The renal papilla is where the renal pyramids meet the minor calyx. Urine flows from the minor calyxes to the major calyxes, which then empty into the renal pelvis. From here, urine flows to the bladder via the ureter. Blood is supplied to the kidney by the renal artery, and blood is removed from the kidney by the renal vein, both of which lie within close proximity of the ureter. The area where these three components associate with the kidney is called the hilus, which is surrounded by adipose tissue in the renal sinus. The renal cortex holds approximately 1.2 million nephrons, which are the functional units of the kidney. Within the kidney, the renal cortex holds the glomerulus, the proximal convoluted tubule, and the distal convoluted tubule. The medulla holds the straight tubular elements in the renal pyramids, which include the loop of Henle, the collecting ducts, and the associated blood vessels, the vasa recta. Blood vessels within the cortex, which are associated with the proximal and distal convoluted tubules, are the peritubular capillaries. The tubule has segmented specialization, and the cells which compose the tubule have varied permeabilities to water, allowing the kidney to retain water and change urine concentration. The kidneys are central to the regulation of water and all major anion and cation concentrations in the body. The nephron consists of the glomerulus, the proximal convoluted tubule, the loop of Henle, and then the distal convoluted tubule. The glomerulus is the site of filtrate production. The proximal convoluted tubule is the most metabolically active and is where 60 to 80% of filtrate volume is reabsorbed. The loop of Henle consists of the thin descending limb and the thick ascending limb, which are involved in the determination of urine concentration. The distal convoluted tubule is involved in the fine-tuning of urine. Both reabsorption and secretion occur here. The collecting ducts connect the nephrons to the ureter via the calyxes, and are involved in electrolyte and fluid balance via reabsorption and excretion. Excretion is controlled by the hormones aldosterone and arginine-vasopressin. Aldosterone increases the rate of water reabsorption in the distal convoluted tubule. Arginine-vasopressin causes more water to be reabsorbed by the collecting ducts. Both these hormones act when the body is in a state of dehydration and needs to retain more water. Urine is formed in a three-step process. Firstly, plasma is filtered in the glomerulus to produce a filtrate composed of water and small molecular weighted solutes. Urine is produced from the filtrate via selective reabsorption of metabolically useful substances from the tubular fluid into the surrounding capillaries, and subsequent active secretion of other toxins back into the tubule from the capillaries. Filtrate consists of water, glucose, amino acids, creatinine, urea, and solutes including sodium, potassium, and bicarbonate. Once filtrate production in the glomerulus has occurred, filtrate flows through the nephron tubules and collecting ducts, resulting in the production of urine. As mentioned previously, the proximal convoluted tubule is the predominant site of reabsorption and secretion within the nephron. Here, 60 to 80% of the filtrate produced is reabsorbed, either actively or passively. Reabsorbed components include sodium, chloride, potassium, glucose, amino acids, and bicarbonate. Secreted molecules include organic and uric acids. The loop of Henle mainly affects the concentration of urine. Water is reabsorbed in the descending limb, and sodium and chloride are reabsorbed in the ascending limb. This results in a higher concentration of waste products, such as urea, in the urine. The distal convoluted tubule is involved in the fine-tuning of urine and carries out final adjustments. Sodium, chloride, and water are actively reabsorbed by tubular cells in exchange for the secretion of potassium and hydrogen ions. The active processes occur due to the action of aldosterone. Water and solute loss is regulated in the collecting system by aldosterone and arginine vasopressin. Reabsorbed molecules include urea, bicarbonate, sodium ions, and water. Secreted molecules include hydrogen and bicarbonate ions. Products excreted in the urine include water, sodium, chloride, bicarbonate, potassium, creatinine, and urea. As mentioned, blood is filtered in the capillaries inside the glomerulus to produce a filtrate. The glomerulus is surrounded by a series of parietal epithelium, known as the Bowman's capsule. Residing between the Bowman's capsule and the glomerulus capillaries, there is an area where filtrate collects before flowing to the proximal convoluted tubule. This is known as the Bowman's space. The afferent arteriole carries blood to the glomerulus, and the efferent arteriole draws blood away from it. These arterioles can constrict and dilate in order to control the rate of filtration. This is called the glomerular filtration rate, and like all blood vessels, the arterioles are lined with endothelial cells. The proximal convoluted tubule has characteristic brush border epithelial cells, presumably to increase the surface area, allowing for greater secretion and reabsorption of solutes and water. Lying between the two arterioles is the distal convoluted tubule. The capillaries are covered in a visceral type of epithelium known as podocytes, which prevent large molecules such as albumin from entering and being filtered by the kidneys. Between the distal convoluted tubule and the glomerulus, there are a group of specialized cells known as the juxtaglomerular apparatus. These include three cell types: granular cells, which release renin; macular denser cells, which detect sodium and chloride passing through the distal convoluted tubule and signal the granular cells to release renin; and finally, extraglomerular mesangial cells, which have an unclear function. The kidney works along with the Renin-Angiotensin-Aldosterone-System to regulate long-term blood pressure. When blood pressure decreases, special cells, the juxtaglomerular cells of the nephron, within the kidney, detect the change due to low fluid volume moving through the nephrons. This stimulates the release of renin into the bloodstream. Renin acts by converting an inactive pre-enzyme, which is released by the liver, angiotensinogen, into its active form of angiotensin 1. Angiotensin-1 has a weak effect on blood pressure. However, to account for larger changes, it is converted to angiotensin-2. This is predominantly carried out in the lungs by angiotensin-converting enzymes. Due to the strength of the hormone, angiotensin-2 can directly act on blood vessels to increase blood pressure. However, its primary function is to stimulate the release of aldosterone, a vasoconstrictor released from the adrenal glands when cells of the zona glomerulosa are stimulated. Aldosterone also affects the baseline filtering activity within the kidneys by acting on the collecting ducts and distal convoluted tubule of the nephron. Aldosterone inserts channels into the cells of the distal convoluted tubule, allowing sodium to move from the filtrate in exchange for potassium and hydrogen. It results in increased water retention within the kidneys due to increased sodium reabsorption and increased potassium secretion, which in turn raises blood pressure. This pathway is initiated in states of dehydration and in response to low blood pressure, and can be interrupted by a number of drugs. These drugs include ACE inhibitors, sartans, which are angiotensin II receptor blockers, and spironolactone, which blocks the action of aldosterone. Angiotensin II is thought to be involved in inappropriate remodeling of the heart following a heart attack, so ACE inhibitors and sartans are often used to lower blood pressure and improve heart attack outcomes.